Welcome to the Cohen Veterans Bioscience webinar series. This is a monthly program sponsored by Cohen Veterans Bioscience for the brain research and systems biology communities. Cohen Veterans Bioscience is a nonprofit research alliance whose mission is to transform research in veterans' mental health through translational research. We are focused on understanding the molecular underpinnings of diseases such as PTSD and TBI, to develop better treatments and diagnostics. Before I introduce today's speaker, let me walk you through some logistics. This program is being recorded and will be archived in the Cohen Veteran Bioscience YouTube channel and on the Cohen Veteran Bioscience website. The presentation will be approximately 45 minutes in length to allow for Q&A afterwards. Please use the Q&A feature on the website to post a question, and we will pose the question to the presenter. Today, our speaker is Dr. Ken Kosik. Dr. Kosik served as professor at the Harvard Medical School from 1996 until 2004, when he became the Harriman Professor and co-director of the Neuroscience Research Institute at the University of California, Santa Barbara. He co-directs the Tau Consortium and co-founded the Learning and the Brain Conference series. He received a Whitaker Health Sciences Award from MIT, a Milton Foundation Award from the Harvard Medical School, the Moore Award from the American Association of Neuropathologists, the Metropolitan Life Award, the Derek Denny Brown Award from the American Neurologic Association, the Zenith and Temple Awards from the Alzheimer's Association, the Ranuel Caputo Medal from the Argentine Society of Neurochemistry, the NASA Group Achievement Award from Neurolab Team, the Premio Aventis from the Accademia Nacional de Medicina Colombia, and the Santa Barbara Innovation Star Award. He co-authored The Alzheimer's Solution, How Today's Care is Failing Millions and How We Can Do Better, and Outsmarting Alzheimer's Disease. His work, including the characterization in Colombia of the largest family in the world with familial Alzheimer's, has appeared in the New York Times, the Wall Street Journal, and the New Yorker, and on BBC, CNN, PBS, and the CBS show 60 Minutes. It is my pleasure to introduce Dr. Kosik, who will discuss the Tau Biology project recently undertaken through the Cohen Veterans Bioscience. Tau plays a crucial role in many neurodegenerative conditions, including the late sequelae of repeated head injury known as the chronic traumatic encephalopathy. Recently, a surge of insights concerning several proteins, including tau, have revealed mechanisms by which they become detached from the surrounding milieu and become vulnerable to the formation of pathologic inclusions. These inclusions take the form of rope-like fibers that eventually strangle neurons. It is my pleasure to introduce Dr. Kosik. Dr. Kosik.
Thank you. Thank you very much. It's really a pleasure to be able to do this webinar for the Cohen Veterans Group. I think that the mission of this group is very, very timely and very relevant to the topic of tau biology, which has been the subject of my research over my entire career. So I have showing you on this first slide a picture of a neurofibrillary tangle, which goes back to the time of Alzheimer. And these tangles are made up of the tau protein. They are one of the two major hallmarks of Alzheimer's disease, the other one being the amyloid plaques. But as we've been learning more and more about the neurofibrillary tangles, it's become quite apparent that they are present in many other conditions. And that's why the biology of tau is so relevant to Cohen veterans, because one of the conditions in which tau becomes abnormal, the protein misfolds, it ends up as these inclusions called neurofibrillary tangles, is chronic traumatic encephalopathy, which is one of the late sequelae of repeated head injury, or what has sometimes been called mild traumatic brain injury, MTBI. So let's go into some depth on this topic. And I think it is really very hard to talk about tau and the conditions that it is, of which it's characteristic, unless we put this problem in the context of the neurodegenerative field more broadly. So here you see three circles. These circles are meant to encompass the mechanisms by which various neurodegenerative conditions operate. And this is a little bit like the blind man feeling the elephant. Pathologists tend to see various neurodegenerative diseases, whether it's Alzheimer's or ALS or Huntington's, by looking at the brain tissue. Clinicians will note the clinical characteristics of these patients that are often very typical, the movement disorders of Huntington's disease, the cognitive and memory impairments in Alzheimer's, and will define the condition on the way the patients present. Over the last decade or 2 decades, gradually we have learned about gene mutations which underlie many of these diseases. And this circle, which is actually the biggest circle, has really created a very fresh way of looking at various neurodegenerative conditions, because the very clean distinctions, or if not clean, at least somewhat, at least conditions that have been considered to be established, are not quite so established when we look at their genetic bases. So that very often mutations do not map cleanly onto one or another condition. There are some mutations that can be associated with both ALS or frontotemporal dementia, just to name one example. So gene mutations have been very helpful, and they have caused us to create perhaps a new designations, a new nosology of these diseases. And I'm now going to fill in these circles, and you can see how complex it gets. So again, the pathologists in the upper circle are looking at the various inclusions, whether they're neurofibrillary tangles, designated NFTs in the upper right corner, all the way down to the amyloid plaques. And many of these inclusions are picked up with antibodies to specific proteins, such as you see TDP43 is one of them on that list. If you go down to the next circle, you see a list of neurodegenerative diseases that all have their characteristic clinical presentations and can often be distinguished from each other by astute clinicians quite effectively. At the bottom of the list, you see CTE, chronic traumatic encephalopathy, which we now know belongs very squarely on this list. And then if we go over to where you saw gene mutations previously, you can see a list of the various genes which harbor mutations associated with the various diseases in the other two circles. And these these genes are grouped together. For instance, over on the, maybe you can see my arrow here, there is a list of genes that are associated with Parkinson's disease. In the middle of the circle, there's a list of genes associated with Alzheimer's that many people are familiar with, such as the amyloid precursor protein and presenilin. Also ApoE4, a risk factor gene. And there are mutations associated with frontotemporal dementia over on the left-hand side of that circle. So we have quite a panoply of genes to work with, and this has provided an extremely rich investigative basis for getting to getting a deeper understanding of these diseases. And here on the next slide, I'm now going to focus in a bit more on the neurofibrillary tangles and tau. And really, what's prominent on this slide are these black boxes. And this is where we stand. There's an awful lot that we don't understand. The genes have been extremely revealing. But as we move from the genes to the underlying cell biology, we still face a black box. And so to go through this slide a little bit, over on the far left, I've listed a couple of ways in which some neurodegenerative diseases, particularly those that involve tau and tangles, are initiated. That is, particular gene mutations can do it. That's why genotype is at the top of the list. Today, we're going to spend a little bit of time talking about chronic traumatic encephalopathy, repeated concussive injury, which also feeds into tangles and represents another important clue about their pathogenesis. In Alzheimer's disease, A beta deposition is maybe a precipitant for neurofibrillary tangle formation. And I have other there, which actually that would be a very, very long list of other causes for neurofibrillary tangles, which include some metabolic conditions and a long, quite a long list of rather rare entities. So all of these disease initiators feed into this black box, which is what happens in the neurons or the astrocytes as they undergo this transformation to form neurofibrillary tangles, the NFTs that you see coming out of that black box. We know that the NFTs are the cell types that are affected. You see down below the NFTs that neurons and astrocytes can both be affected. And the other clue we have is that neurofibrillary tangles seem to be deposited in rather interesting topographies. That is, there are certain anatomical regions of the brain that have a particular predisposition to forming neurofibrillary tangles, the hippocampus, the amygdala, the cerebral cortex, and other areas which are often relatively spared, such as the cerebellum. And then we have an even more mysterious black box, that's why it's a little bit bigger, in which the deposition of tangles in some distribution results in a clinical phenotype. And how we go from the pathology that we see in the brain to these manifestations of the disease, which we'll talk about, is also still rather unclear. One thing we can say is that the clinical phenotypes associated with neurofibrillary tangles are highly variable. There is a huge range of neurologic and psychiatric signs and symptoms associated with many of the various conditions that in which tau is involved. So I'm going to show you a little bit about tau here. This shows you the red bar in the center as O, and here I was told to turn all these things off, and it looks like I was unable to get rid of that one. So here the red bar shows you the length of the tau protein, and it has a very interesting structure. You see R1 through R4. These are repeated regions in which tau will bind to microtubules. This is thought to be one of the main functions of tau. It binds to the microtubules in neurons where it stabilizes them and makes them competent to transport organelles along them like railroad tracks. And tau is, and the R2 there is an alternatively spliced exon. That's why it's in green. It can either be present or not. And at the other end of tau, N1, N2 are two more alternatively spliced exons. You see here that there are many post-translational modifications of tau. That's acetylated, that's what the AC stands for. It can be phosphorylated, that's what the small p stands for. It can be ubiquitinated, is what the UB stands for. And there are other modifications that are not even shown here. So tau is extremely complex with regard to its both alternative splicing and its multitude of post-translational modifications. Now, tau also will harbor a number of mutations. These are huge clues for us as to understand tau pathobiology, and it's littered with mutations. But the mutations do seem to have a sort of characteristic distribution. So you can see here these mutations that are indicated by the amino acids at the particular site, the wild type at the site, and then followed by the amino acid to which it's changed. And you can see that a number of these amino acid changes lie within the microtubule binding region or immediately adjacent to it. So one clue is that something in tau in this region, perhaps related to its microtubule binding properties, or perhaps some biophysical feature of this region is going to be important. The other clue we have here is that you can see 10 is the exon that I referred to previously that was called R2 in the previous slide. Exon 10 corresponds precisely to the second repeat, which is alternatively spliced. And if you can see here that around exon 10, within the introns, are a number of mutations that are called splice site mutations. And all of them do the same thing. They basically increase the tendency for tau to include exon 10 in its final transcript. So as a result of these mutations, the patients have a small excess in the ratio of a 3 repeat to 4 repeat tau. There's increased amounts, sometimes a very small amount of 4 repeat tau. And this means that the actual sequence of tau is completely normal. It's just the ratio of these two isoforms is different. And that problem too, quite mysteriously, will result in a neurodegenerative condition, usually frontotemporal dementia. So we have a couple clues here as to where the problems what might give rise to the phenotypes, a couple of genotypic clues for studying the phenotypes. Mention one more thing very quickly here, that there's one more mutation called A152T. You see that in exon 7 over on the left, which in contrast to all of these other mutations, which are fairly highly penetrant mutations and usually autosomal dominant, this single variant A152 is actually a risk factor mutation, is not a causative mutation. So you can think of it a little bit like ApoE for Alzheimer's, where it's not a causative mutation, but will just simply increase the risk for getting a neurodegenerative condition. Okay, as we progress here, I'm now showing you a list of signs and symptoms of chronic traumatic encephalopathy, which I'm sure the listening audience is quite familiar with, but I do want to spend a couple slides on this because I think this is very important for the mission of the Cohen Veterans Group to try to link this very important and increasingly recognized, perhaps more common than we realize, sequela of head trauma and tau pathobiology. So we still have an enormous amount to learn about CTE. Its clinical picture is highly variable, and I've listed here on this slide the various clinical features of CTE, and you can read these for yourself or when you're reviewing this presentation. But some of the very prominent ones are cognitive impairment and executive dysfunction. Emotional instability is a very important feature. And because of the attention this condition has gotten, we're also aware that such individuals often have suicidal thoughts and act on those thoughts. But it's a very diverse syndrome that can be clinically difficult to diagnose. Now, the pathology is very characteristic, and this is I'm going to show you the next couple of slides from the work of Anne McGee, who has been the leading individual in this area, who's really just made enormous contributions to understanding the pathology of CTE. So this slide is taken from her work, and it shows you some of the gross clinical features of the brain, the postmortem brain when it's removed. There's an arrow there that points to the cavum septum pellucidum, a space in the septum that divides the two ventricles, which is why it's called a cavum. And this is very, very characteristic. You don't always see it, but when you do, it's pretty quite indicative of the condition. You can see here that with my arrow that the frontal and temporal horns are enlarged in the ventricles. There are prominent perivascular spaces in the white matter, particularly the temporal lobe, and a number of additional features in the pathology, which you can see in the final line here, including septal fenestrations, atrophy of the thalamus, hypothalamus, mammillary bodies, thinning of the isthmus of the septum corpus callosum, and interestingly, depigmentation of the locus ceruleus and substantia nigra. We do remember that depigmentation of the substantia nigra is probably responsible for sometimes the Parkinsonian-like features that these patients will have. Again, from the work of Anne McGee, looking at now the microscopic pathology, we have increasingly appreciated that tau is the common denominator that links the CTE with other tau-related neurodegenerative conditions. However, in CTE, the distribution of the tau lesions is very, very characteristic. Often you see the tau lesions at the depths of sulci down here. We see tau lesions along this thin rim of cortex, and these patterns are very, very characteristic. At higher, you can see in the circle below at higher power what a neurofibrillary tangle looks like when it's stained with a tau antibody. And another feature of CTE pathologically is the collection of these tau reactive cells around blood vessels, as you see in the two lower pictures here. And I think that's particularly interesting, a particularly interesting clue, because perhaps the trauma is somehow causing damage. This is just speculative, causing damage to the vessel wall and maybe extravasation of some plasma blood component that might be inducing these tangles. So a lot of what I said is just written down here below the slide. Interestingly, as I mentioned, when talking about the circles, these conditions often overlap, and therefore another protein associated with neurodegenerative conditions, TDP43, is present in many cases of CTE. And finally, one more credit to Anne is she reviewed here the CTE in association with military exposures. It's something we have to pay more attention to, because while the situation with athletes has gotten a lot of attention, it's even more complicated in military settings. So here you can see there have been case reports of CTE occurring in young veterans from Iraq and Afghanistan exposed to blast injuries. Many of the symptoms are typical of CTE, the clinical symptoms, and it is complicated, however, by often having concomitant PTSD. So, of course, the military-related TBIs occur in a huge range of ways, not the more stereotypical ways that it happens in athletics. So there are, of course, recreational activities, physical training practices, falls, motor vehicle accidents. But one we have to, of course, pay a lot of attention to is the exposure to blast injuries. And there too, there's variation. Did the blast occur in an open field? Did it occur inside a building or a motor vehicle? All of these affect how these blast waves will impact the brain. So you can see here the neurodegenerative pathologies that occur after pure military mild TBI remain largely unknown, and it's an important area for investigation. one way to investigate this, which I'm not going to talk about, it's not my area of expertise, is neuroimaging of white matter tracks using diffusion tensor imaging to reveal axonal injury that it might be related to combat. So to get to more of the cell biology and the molecular biology, which is what I do, there's a little bit of literature on trying to model these TBI type conditions. This is one example here where you can look in the upper left-hand corner where a bead has been placed in the brain of a rodent, causing compression of the brain. And you can see when it's compressed that these dendrites here become sort of squiggly looking, which you see down in the lower panel as well. And when this happens, and when there's other models of that have been studied, not just using compression, but using acceleration, deceleration type injuries, it's quite interesting that tau phosphorylation is induced because we know that the tau in the neurofibrillary tangles tends to be very highly phosphorylated. Very often misfolding is induced in these animal models. And of course, that is exactly what's happening to tau as it begins to aggregate into tangles. And even cleavage of the tau protein occurs, which is another feature of tau pathology. And somewhere in that path, whether it's the cleavage products or the misfolded tau protein templating, other misfolding events, will emerge the neurotoxic properties in, say, Alzheimer's disease and CTE and a number of the tau-related conditions. In the color panel down below here, I just want to point out, because this will come up, is that tau, as I mentioned, is a microtubule binding protein, and it tends to be restricted to the axon. Not absolutely, but for the most part in neurons, it's an axonal protein, whereas its counterpart, MAP2, another very homologous microtubule binding protein, tends to be in the somatodendritic compartment. And therefore, these two proteins are segregated, which will, I think, have important ramifications that I'll talk about if there's time. So let's discuss a few of the ideas now about how tau might result in pathology. So here on the upper left-hand corner is a nice, happy-looking microtubule with tau attached to it. And then there's some disease initiation, which you go down to the first part of the figure here, you see tau gets hyperphosphorylated. Then as you go to number 2, this hyperphosphorylation of tau tends to be a trigger for its ubiquitination. Ubiquitination now will target the protein for degradation because all of this hyperphosphorylation may possibly not be so good for the cell. So it's targeted for degradation. And it's also going to be, at this stage, there's probably folding changes that happen in tau as well. So as a result of these changes, while on the one hand it's being targeted toward the proteasome to be degraded, it's also being attached to various chaperones like heat shock protein 70 and CHIP to attempt, and now I may be reading a little bit more into the cell biology than actually is true, but it just seems like it's an attempt perhaps to salvage tau in some way, to refold it, and instead of destroying it, putting it back into the normal biological pathways. So this number 2 here is a very important node. A cell has to decide whether it wants to salvage a protein and reuse it once it has some damage, or to destroy it, to say, okay, I'm just going to give up and I'm going to throw it away in the trash can, which is the proteasome up in number 3. And here comes the big problem. That is, although tau, when the cell decides to throw it away and it's polyubiquitinated, as you see with all these red ovals, for some reason that we don't understand, it fails to get degraded. It just sits and accumulates in the cell. So we start to go down to number 4, where now instead of being degraded, it aggregates into these polymers, and eventually it can, the accumulation of these aggregates will lead to cell death, which you see at the very bottom here. So this is one pathway that we feel we know at least a little bit about. And it gives rise to, on the next slide, here's the same picture, but now it gives rise to two important questions. What are the biophysical properties of tau that predispose it to misfolding and spread? As you know, we also know that tau, once it's misfolded, seems to be able to get out of a cell and move into the adjacent cell. It's an emerging idea that tau may have some prion properties that allow the misfolded protein not only to spread, but once it's in a neighboring cell to template the normal tau in the adjacent cell and induce its misfolding. And then there was the other problem that I identified. Why does the cell not degrade tau as it does other misfolded proteins? Well, so here is another view of tau inclusions. We've known for a long time that by light microscopy, you'd see down here that these are looking, they fill up the cells here. These are pyramidal cells because they have that triangular shape, and tau is accumulating there in these rope-like structures, eventually destroying the cell. This is probably next to it. This pyramidal cell is probably a dead cell, a kind of a ghost tangle. And tau also forms these neuropil threads you see in the middle panel here, where tau not only is accumulating in the cell body, but it's also accumulating in the out in dendrites and also in the axons, where it tends to form these swollen, thickened, what we call dystrophic neurites. It can form also in glial cells. And when you look at the ultrastructure, there's many different shapes that tau can take. This is some beautiful work from Mark Diamond, in which he's shown that tau filaments at this level can have different appearances. But the classical appearance right here is this paired helical filament with an 80 nanometer periodicity in the twisting of it. It's also work from quite a number of years ago by George Perry, who showed that by immunogold labeling, you can label these paired helical filaments here with these gold particles attached to a tau antibody. Now, what we also know in this lower right panel is that we can induce tau filaments in the laboratory, and this is what they look like. Let me talk a little bit about that. So this ability to induce tau filaments gets at the biophysical properties of tau. There is, we know that full-length tau in vitro is highly soluble. It does not self-assemble under physiologic concentrations, even when stored for a long time. But with an activator, and the most potent activator is heparin, but in general, polyanions are activators. Heparin will very rapidly induce this kind of filamentous appearance of tau in a test tube. But other polyanions do too, and one of them, which is of interest to us and to others, given the way the field is moving, is that RNA is a polyanion that can also induce tau assembly. This has been known for a number of years, shown early on by the Mandel cows. And this is going to be, I think, an interesting lead for us, because tau will come into contact with RNA in cells. Tau in the cells will not see heparin. It's not present inside cells. So another feature of tau, which connects it to other neurodegenerative disorders, especially those involving certain proteins connected to ALS, such as TDP-43 and FUS, is that tau is an intrinsically disordered protein. And I sort of convey this idea here by showing on the left, the more triangular appearance, in which proteins are folded in certain ways, and this folding structure sort of locks them into a an energy trough, a low energy state in which they have a certain amount of stability. This is what happened maybe with, say, an enzyme might do this. But tau its conformational landscape is much more spread out, as you see on the right. So it has many, many different conformations, and some of these conformations which could even represent an energy trough, can predispose tau, we suspect, to misfolding. So tau will sample numerous dissimilar conformations, which is important biologically. These different conformations will facilitate its different functions. And at the bottom, you see here that intrinsically disordered proteins are commonly involved in pathology. So other examples of such proteins are amyloid, not just in neurodegeneration, but P53 in the cancer field, Huntington's, synuclein, are all intrinsically disordered proteins. And some of those involved in neurodegeneration, as I mentioned, FUS and TDP43 or RNA binding proteins. There are thousands of RNA binding proteins in the human genome. They have a wide variety of functions shown on this slide. I won't have time to talk about the data today, but we do have emerging data that tau2 is likely to be an RNA binding protein, and thus join this group of known RNA binding proteins, FUS, TDP43, SNRMP, work that has come from the labs of people like Paul Taylor and Steve McKnight. So I want to talk about now for a moment what happens when you have an intrinsically disordered RNA binding protein. What happens in to the biology of a cell under these, in the context of this category of proteins. And by the context, I mean a tendency for these proteins to form RNA granules. These are fascinating structures. We described them in neurons quite a few years ago. The references are shown here. But they've been seen in other systems too. They're present throughout phylogeny. In germ cells of C. elegans, for instance, there are RNA granules. And they have some interesting properties which are shown up at the top, are mentioned at the top here. They are translationally silent, even though they contain mRNAs. Interesting and curious. And even more interesting is that although they move cohesively through cells, in this case neurons, you can see here these little white areas, and I'll show you, I think, in the next slide, that they are moving along. They do so without any membrane around them, as shown in this electron micrograph that we published back in one of these papers. So so how it can be that the contents of this granule remain together while moving remain together, moving cohesively through the cytoplasm without a membrane, has been of interest. And I think the problem has been cracked to some extent in work by Steve McKnight and others, in which he's talked about a liquid-liquid phase-separated state that creates sharp local concentration differences to spatially confine cellular components and control reaction rates without a membranous compartment. That's what we're looking at here. When we saw these things many years ago, we didn't recognize that feature. We saw they were without membranes, but did not understand this phase relationship that has been now become much more apparent and driving a lot of interesting biophysics. So here again is another picture that we published in which RNA granules are moving down a dendrite here. You can just follow these arrows from A through F. They move quite cohesively. We're able to label them with certain characteristic markers and watch them translocate through the neuron. Now, RNA granules are a broad general category. One type of an RNA granule is a stress granule. This is a type of granule described by Paul Anderson, and it has many similar features. That is, again, it moves cohesively through cells. It has no membrane. It's filled with RNA and certain proteins, and is a compartment in which translation is silenced. So we have, and this has been very important, a way you can induce in the lab are these stress granules experimentally is by treating cells with a stressor. Arsenite is a common method by which this is done. So let's talk about this for a moment, talk about cell stress. When a cell undergoes cell stress, it is, it is sensed by a number of kinases, and these kinases will, if you follow along the first arrow here, they'll converge on phosphorylating initiation factor 2A, which will shut down translation initiation. And as a result, you now translation is stalled and you induce this, what I talked about, the translational silencing. Now there's two more arrows here with the kinases, which I have question marks because I think this looks like it could be an interesting direction. We do know from the work of Geraldine Seydoux at Hopkins that phosphorylation is a very important control mechanism over the assembly of RNA granules in C. elegans. And we do, so that's one arrow in which we might try to connect stress, kinases, and RNA granules. And we also know in this next arrow that probably on these neurodegenerative conditions in which tau mutation may be present, or there's head injury, that again, there's cell stress. We know that tau is phosphorylated, and there may be another arrow here that we can try to build some intermediates. Cell stress will also inactivate the mTOR, which will again result in stalled initiation complexes, and their associated RNAs and RNA binding proteins now, once again, as I mentioned, assemble into these foci termed stress granules. And here's the reference to Paul Anderson's work. Roy Parker has also been a very important contributor to this area. Okay, so an amazing thing about tau, a well-known thing about tau is that it is phosphorylated by so many different kinases. I think that remains one of the very big mysteries of tau. Why does tau have so many control elements operating upon it? And it really is something we don't fully understand. It's clearly very precisely regulated in a multiplicity of ways that really has not been elucidated in detail. We know a lot about these kinases and where they phosphorylate, but what the function is of all these different phosphorylation events is quite unknown. I show this because somewhere here, this may relate to the cell stress that's going on. So to further delve into this concept, we have over here a heat shock or cell stress, and around in the periphery of this circle are the many things that happen as a result of cell stress. You get granule formation, and you get the inhibition of translation as well. During translation gets shut down in the presence of heat shock, which is not happening over on the other side of this circle. So stress looks like it's going to be very, very important factor here. That's pretty obvious to say. Of course, if someone is struck in the head or they're carrying a tau mutation, you would think the cells are stressed, but the detailed pathways of what's happening with stress in relation to granules and translation is very important to uncover. So, but we have a problem, which I alluded to a little bit. RNA, you know, as you look at the second paragraph here, tau is axonal. I already talked about that. But RNA, at least in most mature mammalian neurons that are, I say mature because this is not necessarily true in development, and are functioning normally, not if they're injured, RNA tends to be somatodendritic, not in the axon. However, this kind of distinction, this parcellation of separation of RNA and tau is violated when there's injury, and that may bring these two molecules molecules into association and result perhaps in the connection between tau and RNA and misfolding. Now, there is the other thing that I think is fascinating about tau, this is the last part of the talk, is that there remains an unknown link between tau and neuronal excitability. There's all these little hints in the literature, and there's more than these. I put in boxes 3 very nice papers. One by Tim Miller, recently published in Neuron, in which increasing 4R tau will tend to increase the tendency toward seizures in these animals. There's the reference. Another is a paper by Leonard Mucke, in which there's a syndrome called Dravet syndrome, a severe childhood epilepsy, and by deleting tau in this model, in a model of this disease, because we can model it since we know what the mutation is, you can get some benefit. And here, although this is another study, a very recent study, and it's more on Alzheimer's disease, these results may have something to do with the neurofibrillary tangles of AD, because it's very clear that subclinical epileptiform activity is much more common in AD patients than in controls. And so I think we know so little about the relationship between tau and neuronal excitability. Tau has been so locked into this function of its role of binding to microtubules that perhaps we've overlooked some role for tau in controlling sodium channels or some other facet of tau which is related to neuronal excitability. And these data suggest we should be looking there. And we are. So we have recently, through some funding through DARPA actually, have developed been using multi-electrode arrays here down in this dish, and in collaboration with a physicist in our campus named Paul Hansma, built this neural circuit probe in which we can record from neurons on the multi-electrode array, and do so in the presence of, say, induced pluripotent stem cells that harbor tau mutations, or also create perhaps simulations of injury in the dish and look at the physiology. In doing this, we've actually already have an interesting way to approach this problem using this model system. We can actually detect what we call a propagation signal. That is, there's a neuron, say, over this electrode, and it's sending an axon down here, and we can detect this pathway indicated by the red dots by, if you go over to the far end, looking at these deflections here that happen with sub-millisecond time delays from as you go from each electrode to the next electrode, and are instantiated numerous times, thousands of times, as the signal goes down this axon. So since we're really have implicated tau and axons and perhaps injury to axons, I think this electrophysiological method may turn out to be an informative model. And we can use that neural circuit probe, that pipette coming down from above, to actually drop TTX on a single neuron and silence it, which is a further proof that we're looking at axonal pathway. Well, I think I've probably talked for enough time, so I want to just close by showing you our group. The bottom group is more of the molecular biology and cell biology people. On some of the work we're doing on looking at the biophysics of tau, we're collaborating with a biophysicist on our campus named Song-Hee Han, over in the picture in the lower right. And then we have another more broad campus group that includes a number of people from the physics department. I mentioned Paul Hansma here as well, and Gene Carlson, and some others who are helping us to analyze the data on the multi-electrode arrays and to build some of those devices. So thank you very much.
Thank you, Dr. Kosik. And now we'll wait a second and see if we get some questions. And if so, we'll address the questions to you. I'm going to just give it a little longer. I don't see any questions, so I just want to thank you for a very, very interesting presentation. And if we do get any questions once we sign off, I'll make sure you get them.
Thank you very much.
Thank you very much. Bye-bye.
Bye.